Triple sugar iron agar is useful for the identification of gram-negative bacilli. TSI tests for several things. It tests for hydrogen sulfide production, glucose fermentation, and whether the organism can utilize sucrose or lactose as a carbon source. Hydrogen sulfide production and fermentation are both anaerobic processes. TSI is used to screen for enteric pathogens. Enteric pathogens tend to produce hydrogen sulfide and not use lactose or sucrose, so they give a reaction pattern on TSI that helps determine if the organism is a potential pathogen or not. Most of the enteric flora come from the family Enterobacteriaceae. Since hydrogen sulfide production and fermentation are anaerobic processes, we're going to have to inoculate media in such a way to provide anaerobiosis. You will use a sterile inoculating needle to select the colonies for testing, and then you will stab the butt of the TSI with the inoculating needle. So you will take the needle, stab through the slant all the way to the bottom of the tube. This area will be anaerobic so that fermentation and hydrogen sulfide production can occur. Then as you withdraw the needle, you want to streak the slant. The tube is incubated for 18 to 24 hours at 37 degrees centigrade. After incubation, you'll need to interpret the results. There are many different results possible. You have two different tests going on in this particular tube. As you can see, all different colors of media can occur. Let's look first at hydrogen sulfide production. You'll remember that anaerobic organisms use an inorganic molecule as their final electron acceptor. Some organisms use sulfur as that final electron acceptor, and instead of producing H2O like aerobes do, they produce H2S. Hydrogen sulfide will react with ferrous sulfate, which is incorporated in the TSI media, to make ferrous sulfide. Ferrous sulfide is a black precipitate. So you will look for a black precipitate in the butt of the tube. That is the place where anaerobic processes can occur. So in looking at this tube, we have this black butt. That's positive for hydrogen sulfide, and no blackening in the butt of the tube is negative for hydrogen sulfide production. The other test in the TSI involves sugar metabolism. There are three sugars in TSI: glucose, sucrose, and lactose, and these would be the primary carbon sources for microorganisms. You'll remember that glucose is the best utilized, easiest utilized of all of the sugars, and that organisms are geared to use glucose first, and only use glucose. Once the glucose has been consumed, then the organism will induce operons if they're available. If they have a lactose operon or a sucrose operon, the presence of lactose and sucrose in the media will induce those operons so that those sugars can be used. There's 10 times more sucrose and lactose in the media than glucose, so glucose is very quickly consumed by the organism. At that point, the cell must metabolize some other carbon source. If it has the operons for lactose and sucrose, it can induce those and begin using those sugars. If it does not have those operons, the only other carbon source in the media is protein. Triple sugar iron agar contains phenol red. This is a pH indicator. Under acid conditions, phenol red turns yellow. The end product of sugar metabolism is acid. Glucose is fermented in the first few hours and will turn the tube yellow if the organism ferments glucose. Once the glucose is gone, the organism must utilize some other carbon source. If the organism can use lactose and or sucrose, it will do so. More acid will be produced and the entire tube will remain yellow. If protein is used, the first thing that has to happen is you have to pull the amine group off. That's deamination, and that results in the alkaline end product ammonia. Under alkaline conditions, phenol red turns red. Proteins can only be used aerobically, so this reaction occurs only on the slant. Glucose fermentation equals acid produced. That's going to give you yellow agar. When you're reporting this, you will use a capital A. Fermentation may also result in hydrogen gas being produced. If that happens, you'll see bubbles in the agar, or the agar will be pushed up from the bottom of the tube. Sucrose and or lactose utilization also yields acid production and yellow agar. But if protein is utilized, you get alkaline end products and you get red agar. The capital letter K is used to indicate alkaline conditions. Here we see a tube that's all yellow. The yellow slant tells us that the organism was able to use either sucrose and or lactose to maintain acid conditions in the tube. The fact that the butt is yellow indicates that glucose was fermented. And here we have the agar pushed up from the bottom of the tube, indicating gas was produced. That's an awful lot to write down, so we use the shorthand A/AG. This is the slant reaction, and this is the butt reaction. Here's another tube. This one has a red slant. So the red slant indicates that protein was used. Sucrose and lactose could not be used by this organism. The utilization of protein gave us alkaline conditions that can only occur aerobically, and we only have aerobic conditions on the slant. But the butt is yellow. That indicates that glucose was fermented. We also have the gas bubble. This would be reported as K/AG. K for the alkaline slant and Ag for the acid and gas produced in the butt. This one has a red slant and what may at first glance look like it is a yellow butt. The red slant indicates that protein is used, so sucrose and lactose could not be used. But the butt is really red. Actually, there's been no change in the butt. The agar started this color. If you ever have one of these reactions, you might want to compare the tube to an uninoculated tube. It's easy to read this the wrong way. This would be reported as K/K. This would indicate that this was a non-fermenting organism or an aerobic organism. In this tube, we can clearly see the yellow slant, telling us that sucrose and or lactose was used. But the butt is so heavily covered with hydrogen sulfide that we really can't tell if the butt is yellow or red. This is reported as A/H2S to indicate that we had acid conditions on the slant, but in the butt we had such excessive hydrogen sulfide production we could not read the sugar reactions. When interpreting the sugar metabolism results, the slant reaction tells you whether sucrose or lactose was used. If the slant is yellow, they were. If the slant is red, protein was used. The butt reaction tells you whether glucose was fermented. A yellow butt indicates fermentation, a red butt or no change, no fermentation. Hydrogen sulfide production also only occurs in the butt because it's an anaerobic process. So the TSI is going to have two reporting lines, one for hydrogen sulfide, either positive or negative, and the other for the sugar metabolism, which should be reported as the slant over the butt reaction, things like A/A, K/A, A/AG.